Thank you very much for the organizers for having me present my work. My name is Alex. I'm an HFSP, or I was an HFSP long-term fellow in the lab of Charlie Rice at Rockefeller University. I'll try to use the laser pointer from PowerPoint so you can see it on both screens. And my project was revolving around innate immune memory following a respiratory viral infection, and the model we used was SARS-CoV-2. So I want to start out quite broad by talking about what the immune system is and how it's organized very broadly. As many of you know, probably you have roughly you can divide into two branches. One is the innate branch of immune responses that consists of innate immune cells such as macrophages and others. And innate immune response is characterized by very rapid responses that are initiated via pattern recognition. So patterns are like biological structures that are shared by a couple of microbes or pathogens, such as, for example, double-stranded RNA for viruses, which is usually not host-derived. So this allows a very rapid and non-specific but broad response that usually was thought to not result into memory. So after you get an innate response, this usually tapers off and that's it. Whereas in the adaptive arm of immunity, most famously known T and B cells, the activation takes a bit longer, like up to 7 days in humans and mice. But the basis is a very specific antigen recognition, that it's usually a short peptide, which allows highly specific immune responses that are very robust and also allow memory formation that can last for years and decades. However, this is also like one of the caveats of adaptive immunity, because if you're highly specific, you're kind of prone, for example, in the case of a virus, if the virus mutates, it can happen that your antibodies that are produced by B cells do not work properly anymore or not work at all anymore. So you kind of need to go through a new round of infection to eventually acquire memory again. So in the recent years, there has been a new field that has been emerging, which is called innate immune memory. And what this is that it kind of changed our view of how innate responses result in memory. Because innate responses, usually if you have a primary activation, you get some response. It's just an arbitrary unit here. Then this kind of tapers off. You enter homeostasis again, so no response. And once an innate immune cell, such as, for example, a macrophage, encounters another pathogen or another foreign pattern, it will respond differently. And here I depicted that in this case, the cell would respond better. And the difference between those two peaks of response in primary and secondary activation is what we call innate immune response. And through extensive work in the last 10 years, probably what's become very clear is that the basis of this innate immune memory is epigenetic remodeling. So after the first encounter of a pattern or a pathogen, those cells kind of remain or retain an epigenetic scar, so their chromatin is altered that allows an altered secondary response. And why do we care about innate immune memory? I think all of us know too well now about the concept of herd immunity, because if you have, for example, a new pathogen that's encountering a naive population with the right properties of the pathogen, these pathogens in blue are all naive individuals. If one is infected and this pathogen can rapidly spread, this will result in a huge overall health burden of the population and also a huge burden for economies. Whereas if you have a population where some of the individuals here depicted in orange are more resistant or resistant to developing a severe disease, this pathogen is not that much of a burden anymore for the overall population. And kind of the broad question we wanted to ask, or what we hypothesized, is can we actually, or can innate immune memory, if we understand this phenomenon better, be used to bolster or enable new strategies to kind of accelerate achieving herd immunity, or also increase, for example, pandemic preparedness, because this type of memory can potentially be functional for even unknown pathogens because it's very broad. So the question was really innate immune memory in the context of commonly circulating respiratory viruses, or how do past infections shape future infections with unrelated viruses. And the model we used was a mouse infection model using a mouse-adapted strain of SARS-CoV-2. So we infect those mice and let them recover for 30 days. So that's the model I've been using and will use throughout my talk. During acute SARS-2 infection, those mice display severe body weight loss in the acute phase, but then by day 30, they kind of regain or recover from this body weight loss. And also, if we look at lung histology, so what you see here in the first 2 slides, in the first row is acutely infected animals. The brown dots is SARS-2 antigen, so the mice are infected, but then in naive and recovered mice, this antigen is absent, so they are also virus-free, which is quantified here on the right. And also, if we, for example, quantify RNA with more sensitive methods like qPCR, those mice are also negative. So we definitely know that those mice are healthy again after those 30 days. So what we did now is to kind of assess whether there is potential innate memory formation. We did an epigenetic assay on a single cell level, because I mentioned that the basis of innate immune memory is epigenetic remodeling. So we harvested lavage fluids, so all airway-resident immune cells of naive and recovered animals, and then we performed single nuclei sequencing and looking at the chromatin landscape. And what we found is, so this is basically this is a human plot that shows both naive and recovered samples in one plot, and we identified all kind of cell types that we would expect, like macrophages or T cells. But already here, I think it was interesting to see that those macrophages, so this innate immune cell in the lung, they cluster into 2 distinct subsets, and I named them cluster 1 and 2. These myeloid cells also have macrophage-like properties. They're probably from the circulation, but they're also putative macrophages. So now if we color code those cells by sample origin, and in black throughout the presentation I will show naive cells, and in red recovered, I think you can immediately appreciate that this cluster here, which corresponds to cluster 2, is almost exclusively recovered cells, whereas macrophage cluster 1 up here is kind of a mixed bag. So this already, just by an unbiased analysis, shows you that recovered macrophages somewhat look different on a chromatin landscape. But this also comes with a caveat of analysis, because if we now in cluster 2 wanted to ask, okay, what's the difference between naive and recovered, there's basically no comparison to naive. So we took this whole subset of putative macrophages, including the myeloid cells, and then kind of reanalyzed these cells as one and asked, okay, what's now the difference between naive and recovered on a chromatin level? And so this is just one piece of analysis we did, and this is basically analyzing which transcription factor motifs are increased, which accessibility is increased in recovered animals. So on the right is transcription factor motifs that are more open or more accessible on a chromatin level in recovered animals. And probably some of you recognize these transcription factors such as STAT1, STAT2, and IRFs, because they're all part of antiviral innate immune response pathways. So this suggests that in recovered macrophages, those loci that respond to antiviral innate signaling are more open. So I just want to, because I also wanted to briefly introduce again, as a reminder of immunology courses, antiviral innate immune response, how it works, because it will be important for my follow-up experiments. So basically, innate immune response is type 1 interferon response. And how this works is if you have a cell, and in the cytoplasm of this cell, you would have a viral RNA or poly-IC, which is a synthetic mimic of double-stranded RNA, which I will use later in experiments. This foreign pattern is recognized by RIG-I or MDA5, and then via maps on the mitochondria leads to phosphorylation and nuclear translocation of IRF3, so one of the transcription factors we found previously. IRF3 goes to the nucleus, induces type 1 interferons that are secreted, and then signal via the type 1 interferon receptor that's present on every cell in the body to then initiate this transcription factor complex consisting of STAT1, STAT2, and IRFs to go to the nucleus and induce the expression of hundreds of antiviral ISGs, or interferon-stimulated genes. And so our question now was, okay, so we found this increased chromatin accessibility of those IRFs, but does this actually also alter like secondary responses? And to that question, I did an ex vivo experiment. So again, infected mice, let them recover, and then you isolate the airway-resident macrophages and plate them in cell culture, and I stimulate them with different patterns of pathogens and then measure gene induction via qPCR. And what we found is, it looks complicated on first glimpse, but it's quite simple. So on top you have 5 interferon-stimulated genes, so antiviral genes, and on the bottom is 5 more general inflammatory genes. Then you have always 3 samples of naive, 3 samples of recovered, and then it's different stimuli for different innate immune response pathways. And for example, what's quite clear by eye is if you look at poly-IC, so kind of mimicking a viral stimulation, you can see that those recovered cells, they really hyper-induce all these ISGs, suggesting that those cells are actually really mounting functional antiviral memory if they experience another virus, basically. This is also true if we, so kind of in a more functional assay, if we infect those cells with a reporter virus, so this is a GFP-labeled virus that it's called VSV, you can see that in naive cells, there is more virus-positive cells than in recovered cells, so kind of proving that this gene expression changes actually functionally translate to more resistance. But then usually the next question is, okay, this is all in vitro, but is this like of actual physiological relevance? So what we then kind of set out to do is to go back in vivo and challenge our SARS-2 recovered mice with a non-related pathogen. And this non-related pathogen that we chose is influenza A virus strain PR8, which is also a mouse model of influenza H1N1 infection. So basically the setup is infect mice with SARS-2, let them recover for 30 days. And then here I challenge them with a sublethal dose of PR8 and then monitor body weight loss, which is a standard readout in this infection model. And in red is now the SARS-2 PR8 infected animals. So they are recovered and infected, and in blue is naive and infected. And you can appreciate that the mice that have seen SARS-2 before, so that have putative memory, they actually do much better in terms of body weight loss. So it seems that past SARS-2 infection kind of benefits those mice if they were encountering influenza A infection. So can we link this to actually macrophage memory, because it could be completely different observations or phenotypes. So we did 2 experiments to kind of get at this point, and one was to, after we had mice recovered from SARS-2 for 30 days, you treat them with clodronate, which is a depleting agent that depletes all macrophages. So basically we kill off those memory cells in the lung, and then we infect them with PR8. And what we found is, it's a bit of a busy graph, but I think it's quite simple at the end. The full symbols are the reference groups. So in full circles is recovered and PR8 infected or influenza infected. In full blue squares is naive and influenza infected. So you get the difference that I've shown you before. So mice that are recovered and PR8 infected do better. But then if you look at the empty symbols, which is the depleted mice, first of all, they do worse because the mice don't have macrophages, which we expect. But then in those groups, this difference is also lost. So this means that you need macrophages to actually get this difference in body weight loss. So that macrophages are basically necessary to have this memory response. In a second approach, we took the macrophages from recovered animals and then briefly cultured them in vitro and then transferred them into naive recipient mice. So those mice have never seen a pathogen, but just get recovered or naive macrophages. And on top, we also depleted those mice of T cells to, again, like kind of rule out any adaptive responses that could kind of contribute. And when we infect those mice with PR8, the difference is a bit smaller, but I think it's there and reproducible. So in red, if mice received recovered macrophages, they do much better or lose significantly less body weight loss than mice that have received naive macrophages. So kind of showing that macrophages are not only necessary, but also sufficient to kind of ameliorate this secondary or unrelated viral infection. So now if we do like a more drastic model, such as lethal infection with PR8, could be that this memory is not sufficient, but in our case, it turns out it still is. So if you do a survival plot of a lethal infection model, you see that by day 10, all naive influenza-infected mice succumb to disease, whereas the recovered mice are all alive at this point. How does PR8 cause lethality? This is a hyperinflammatory response. So it's not necessarily the virus that kills the host, but kind of the overshooting immune response in the acute phase that is detrimental for the host and then eventually leads to death. So we looked at, for example, neutrophils in the airway. So during the course of infection from day 0 to day 7, we see that in single infected animals, there is significantly more neutrophils coming into the lung. And this also at day 5, so where there's still all mice alive, this correlates that naive infected animals, they have like highly elevated cytokine levels in the airway compared to recovered PR8-infected animals. So this leads me to my conclusion slide, or to kind of the summary. What we found is that if you have a primary infection with SARS-CoV-2 and the mice recover, you kind of in the airway, the macrophages retain innate immune memory that is antigen independent and allows pathogen cross-protection. And this is achieved via increased antiviral activity and interferon response in those macrophages that then downstream during secondary influenza A virus infection results in reduced disease severity, less cytokine storm, and neutrophil recruitment. And with that, I'd like to end. If you got interested, please feel free to check out the first version of the manuscript on bioRxiv. I'd like to thank my mentor, Charlie Rice, the whole Rice Lab, and also Steve at Cornell, who was a very important collaborator in this work. And thank you for your attention and questions.
Yeah, hi, Alex. Very nice work. And so I might have missed here.
Yeah.
Oh, wow. All right, I'll be quick. So I might have missed, how are you making sure that you have completely ruled out T-cell activations and responses because protection from T-cell responses? And the other one was, how universal is it? Do you think this is going to be something common among other kinds of infection, viral infections?
Yeah. So for the T cells, I didn't show this. What we also did is we depleted. So after SARS-2 infection, you do have a lot of memory T cells in the lung. But we also did depletion experiments where we depleted those cells with antibodies and then infect animals, and we still get the same phenotype. So I do think there's little to no contribution of T cell responses in our model. And to the second question, so I think this phenomenon is not necessarily new, but in the context of like respiratory viruses, I think it's like a bit of a black box where we don't know that much anymore. But one thing I can share is that when I swap them, when I do PR8 before or influenza before and then SARS-2, I still see memory. But that's the only combinations I did so far.
I really enjoyed your talk, but I wanted to maybe like push back on one claim, since I think it's an HFSP conference, so we're here to do that. You said at the beginning that sort of in the field, there's kind of this like belief that the sort of the enhancement of the second wave is due to like an epigenetic remodeling.
Yeah.
And I guess I just want to know, like, how sure are we really that that's the case? Because if that's a claim that's based on, say, for instance, like the ATTAC-seq, which is measuring accessibility, like you could of course have changes in accessibility due to like true epigenetic changes like methylation and histone modification, but it could also be that just like you're upregulating these transcription factors, and the transcription factors make the DNA more accessible because it's more transcribed. So in essence, like, could it be also that the memory is encoded in the proteome rather than being encoded in the epigenome? And do we really know? Do we have experiments that disambiguate between those two possibilities?
Yeah. I think we don't really know, and it might be context dependent, which is not the best answer, I think. But in this model, what we also did is we did cut and run for histone marks, like what you suggested. And we do find, if we, for example, look at H3K4 monomethylation, which is like a mark for poised transcription, this is strongly enriched in recovered cells compared to naive. But again, it doesn't directly address your point, I think. Like I can also say that on RNA level, those cells look quite similar if they're not re-stimulated. But again, I think we don't have in-depth mechanistic data, because those cells, if you take them out, if you keep them in culture for a prolonged time, they also change their phenotype. So I also feel like they lose memory once you culture them in 10% FPS, etc. So it could also be that it's an external factor that kind of maintains the memory in vivo, right? That's derived from a different cell. But I think that's future work to be done, I think.
The answer is always proteomics.
Thanks for your talk. I was just wondering, on the level of individual mice, do you see any correlations between like the severity of the initial infection and how they deal in the second infection?
No, we don't. So short answer, I cannot quantify viral load during SARS-2 infection because it's quite locally restricted, so I cannot sample the mice. But if I just look at minimum body weight during SARS-2 and minimum body weight later, there is no correlation.
Thank you.
Yeah, very nice talk indeed. I was wondering about the long-term stability of this macrophage adaptation. Like, do we have any elements, or do you plan on studying that at some point?
Yeah, I think that's also a recurring question that we have. I always refuse to do it because it's also a BSL-3 experience, so it's quite cumbersome because they have to be in high containment. But so I did 60 days post-infection. Memory still works fine. I also did one experiment where I waited 200 days, so like 9 months, and then the memory was gone. So somewhere in between, it's lost. And I think we don't know yet. I think also in a real-world context, if we would think about humans constantly experience PAMs, whereas mice, they're like really, once they recover, they are kind of pathogen-free again. So it's not, I don't know, probably you need always like constant stimulation to kind of keep it going, I would assume, but I don't know.
Okay, thanks.
Hi, thanks for the talk. So it seems from your results that these cells are really rewiring their metabolism to respond to viral infections specifically. So do you see a trade-off where they respond less effectively to bacterial or fungal infections?
I think that's something I want to look into next. I think the lab is historically not set up for bacterial or fungal infections, but I do think that, like, I show a beneficial effect, but I think there might very well be like a scenario where there's no benefit. And especially like for flu, for example, it has been shown that usually flu in humans makes you more susceptible to bacterial infections. And it could also be that this kind of skewing towards innate antiviral immune responses kind of makes you more prone or is detrimental, for example, in a secondary allergy model or some type 2 immune response. But I don't know yet.
Thank you.
Thank you.
Okay. Thank you again for a brilliant talk. Very relevant.